Imagine trillions of microbes throwing a biochemical rave in your gut, fermenting fibers, breaking down proteins, making postbiotics like it's their job, which actually it is, and then sending their metabolic party favors directly into your bloodstream. That's the gut-blood axis in action, a molecular relay system where what your microbes eat, poop out, or secrete eventually winds up influencing your brain, heart, liver, skin, and immune system. It's no longer news that we are hosts to vast microbial populations, but what's blowing open the doors of modern medicine is the realization that their chemical leftovers travel and often shape our long-term health, especially how fast or gracefully we age. Let's start with short-chain fatty acids, SCFAs, the A-listers of microbial metabolites. When your gut bacteria feast on fiber, they produce SCFAs like butyrate, acetate, and propionate, which are then absorbed into the bloodstream. These molecules are more than just gut fuel. Butyrate, for example, reaches the liver and circulates through blood vessels, where it acts as an anti-inflammatory agent, helps regulate insulin sensitivity, and even promotes histone acetylation, nudging gene expression toward a younger, less inflammatory state. Some researchers now call SCFAs postbiotic hormones because of their systemic reach. Translation: your gut microbes are basically tiny pharmaceutical factories pumping out epigenetic influencers that happen to love kale and legumes. Then we have indole derivatives, metabolites produced when gut microbes digest the amino acid tryptophan. These include indole 3, propionic acid, IPA, indole 3, lactic acid, and others that ride the bloodstream like molecular hitchhikers, calming immune responses and enhancing the integrity of the blood-brain barrier. IPA is a particularly flashy compound. It's a potent antioxidant and has been linked to reduced risk of cognitive decline, better vascular health, and increased longevity in animal models. It's not a pharmaceutical, but it behaves like one, sneaking through blood vessels and whispering anti-aging messages wherever it goes. But not everything that flows from the gut into the blood is a gift. Enter lipopolysaccharides, LPS, components of the outer membrane of gram-negative bacteria. When the gut barrier is compromised, LPS leaks into the bloodstream, where it triggers immune alarms, elevates TNF-alpha, and contributes to metabolic endotoxemia. Chronic LPS exposure is a known driver of systemic inflammation, insulin resistance, and even cardiovascular aging. The same goes for TMEO, trimethylamine N-oxide, a metabolite created when gut bacteria digest choline from eggs, red meat, and fish, and which is then oxidized in the liver. High blood levels of TMEO have been linked to arterial stiffness, oxidative stress, and increased cardiovascular risk. But as with most things microbial, the story isn't black and white. Some studies suggest the context and dose of TMEO may be key, and blanket demonization might be premature. The immune system is another major player in the gut-blood axis. Gut microbes train immune cells, and their metabolites influence systemic immunity. SCFAs promote regulatory T-cell expansion, which keeps inflammation in check throughout the body, while other metabolites influence macrophage polarization, shifting them between pro-inflammatory and anti-inflammatory modes. The blood becomes a kind of immunological courier, delivering microbe-mediated programming to distant tissues like joints, lungs, or skin. In aging, where immune function tends to become both hyperreactive and sluggish, a paradox known as immunosenescence, this microbial tuning could either help reboot balance or deepen dysfunction. In fact, many hallmarks of aging, mitochondrial decline, cellular senescence, DNA damage, are now believed to be influenced in part by microbiome blood interactions. Mitochondria are especially vulnerable to oxidative stress, and here again, microbial metabolites like butyrate and IPA step in, enhancing mitochondrial biogenesis and buffering against ROS. Other microbial products can modify NAD+ metabolism, regulate mTOR signaling, or impact AMPK activity, effectively linking dietary patterns to intracellular aging clocks via microbial intermediaries. And let's not forget the neuroendocrine axis, where gut microbes modulate serotonin production, stress hormone regulation, and even blood levels of BDNF, linking digestive activity to brain function and mood. You might feel like you're craving chocolate or feeling inexplicably anxious, but part of the signal could be coming from a community of bacteria whose metabolic gossip has just entered your bloodstream and tickled your hypothalamus. The emerging truth is this: Your blood is not just a reflection of your liver or pancreas, it's a microbial echo chamber. What your gut microbes eat, excrete, or modulate often ends up transforming the chemical landscape of your circulation, for better or worse. In this sense, systemic aging is not only a cellular process, it's also a microbial drama, one in which your daily choices about food, stress, and antibiotics may be shaping your health trajectory via the molecules that microbes inject into your bloodstream. So, if you're serious about living longer, clearer, and calmer, don't just look at your cholesterol or fasting glucose. Ask what your gut bacteria are whispering into your blood, because the molecules floating through your veins may carry the signature not only of your last meal, but also of your future biological age.